Thank you for the kind introduction, and I really appreciate the opportunity to be able to attend this conference. I've been learning a lot about exciting research going on in the field of computational biology. So my name is Haonan. I'm currently a postdoc at the Lawrence Livermore National Lab. Just a disclaimer, my training was mostly in statistics and probabilistic modeling. So I apologize in advance if I'm not able to elaborate more in details the biological side of things. And nevertheless, I'm excited to present our work that to use a deep active learning framework to efficiently pinpoint promising double gene knockdown experiments for therapeutic treatment. So as we all kind of experience in our more recent memories, as we enter into 21st century, there have been increasing number of viral outbreaks, which really affect a population globally. So there's have been a rising urgent need to develop pre-positioned countermeasures against such emerging threats. And there have been many approaches that can be deployed to counter viruses. Meanwhile, I think the host targeting approaches are still an area that's hugely underexplored, and they have been shown in previous work that this indeed could be a viable option. And however, due to viral replications and mutation rates, typically if we develop a drug that's only targeted a single site, the viruses can develop a resistance to the drug over time, which motivate this work where we want to primarily focus on the setting where we want to identify a possible double gene knockdown treatments. And then the main question we want to address is that can we identify those promising gene pairs for knockdown experiment without exhaust search through the entire combinatorial space? So before we deploy this our methods into any real experiments, we would like to first do a proof of concept on existing data sets to see whether this is indeed possible. So in this case, we use data sets that collected from the Kogan's lab at UCSF. In a nutshell, this data set consists of 356 by 356 matrix, where the row and columns corresponding to the pre-selected genes for double knockdowns, and the value of the matrix corresponding to viral replication. So smaller value of the matrices means the combined double knockdown indeed has some suppressive feature for the HIV viruses. So now our goal here would be, is it possible to identify the gene pairs or like entries with the matrix with the smallest value without necessarily query the entire matrix? So before I dive into a little bit more detail into our framework, I just want to give an overall flow chart of the entire work. So in the left figure here, we first, because to kind of reduce the competition complexity of the problem, we have to bring like external information. So in our case here, we use a spoke knowledge graph, which stands for Scalable Precision Medicine over Open Knowledge Engine. And it's really a database of databases where it contains over like million various different nodes, including genes, biological processes, and it contains multiple different type of addresses, such as downregulate or upregulate. And now we're going to use a graph neural network, try to do a self-supervised training on this existing knowledge graph to learn meaningful embeddings that summarize the local topology of those genes. And then once we obtain those embeddings, we'll feed into our iterative process. So to start our iterative process initially, because we don't have any observations, so we just simply do a cold start by randomly select gene pairs from the matrices to uncover. Now, once we observe those matrices, we can fine-tune both our embeddings and also predictive model on those observed entries. And then from there, we can forward predict onto unobserved entries of the matrix, and then pass this through our acquisition function, which I'll discuss a little bit later, to recommend what's the next set of experiment to examine. And this whole process will repeat recurrently until we hit certain experimental budget. So one of the main component of our model, the predict model, is the graph neural network. So in this case, because in the spoke we have a heterogeneous graph, we have like nodes and edges of different kinds. So we use relational graph neural networks where similar to like multi-head attentions, where we're going to have a different convolution weights to combine those different edge type for each node to aggregate information that capture the local topologies from the knowledge graph. And then before I dive a little bit more into the details acquisition function and our results, I'll just introduce like few light notations. So in general, I'm going to denote the embedding for gene I as xi, and then the interaction matrix of interest will be denoted as y, with entries denoted as yij. And then I'm going to denote the predicted interaction matrix by the model as a y hat with entries yij. And as I preluded to, for this setting, we're primarily interested in like whether we can identify like entries with a smaller value. So the main evaluation metric we use is we refer to as a coverage. So which essentially is the fraction of the top 400 genes that we have covered so far. And then this will be in the cumulative setting. So as I alluded to, one of the very important ingredients of this framework is how we decide the acquisition strategy. How do we select the next set of genes that we're going to recommend? So in general, in the literature of active learning or reinforcement learning, there's this trade-off between exploration and exploitation. So exploitation means that essentially we have a predictive model that we simply just pick up the genes that has the best performance predicted by model, but that might has limitations, as maybe the model will highly zoom in on only a certain like subset of genes, which remain like large, which leave the large like remaining matrix unexplored. However, on the other end, if we only focus on exploring as diverse set of genes as possible, this might be very unfriendly, like competition friendly in terms of budget wise. So the main goal here is really try to strike a balance between this exploitation and exploration. So for our setting, we actually have like two settings where we simply has a one base model, and then the other setting we have an ensemble model where we train 20 like same model with different initialization in parallel. So this gives us like a better improvement performance as we can ensemble those predictions together. So in terms of acquisition strategy-wise, the first one would be just a simple greedy strategy, which is really focused on exploitation, where we only select the gene pairs based on the predictive values. And then the next set of acquisition strategy focused on exploration is so-called a batch method in the literature. And then the other is the maximum variance, where for the batch, it's simply just identify the genes that with the maximum magnitude in the gene embedding space, which actually is related in the literature of the like linear optimal experiment design to the information gain criteria. And then another exploration based approach will be we refer to as maximum variance. That simply select the gene pairs where we see most variations among the different ensemble models. And then one approach we utilize try to balance the exploration is we refer to as optimism. So we try to use quantile information from the ensemble predictions. So you can think of this as really try to strike a balance between the greedy approach and also the maximum variance approach. And also we use a few other like model agnostic acquisition functions as like benchmark. So the first one would be a simple uniform random sampling, random selection. And the second one is based on the degree. So we will sample the gene pairs that with the larger the outer degree those nodes are, then the more likely they will be selected by our sampling scheme. And the last one I refer to is called path-based random sampling. So in this one, we will just simply compute the shortest distance given a pair of genes, and then we will simply more likely to sample the nodes that's close based on the knowledge graph. So this is like really based on the intuition that maybe the genes that are actually close on the knowledge graph are more likely to have a synergetic effects. And then in the actual experiment, like when we do the sequential setting, we're going to run our iterative step by a 16 step. And then each step will query about 400 entries of the matrix. So at the terminal phase, we roughly observe about 10% of the entire matrix space. And then this is a result that we evaluate using the coverage at 400 that I described earlier. And as you can see here, both actually the greedy and the optimism approach perform really well. And then we're able to uncover about 92% of the top interaction entries, while only observe less than 6.3% of the matrix. While I think it's worth noting that while greedy does seem competitive results, but optimism does show, like especially in the later run, the benefits of more exploration, that it has a slight edge over the just simple greedy approach. And then we also performed like a sequence of ablation study, try to really understand like what really make this type of approach successful. So one is that we want to compare the base model compared to ensemble model, like to see whether is ensemble really helpful in this setting. And also we want to understand whether the initial self-supervised training on the knowledge graph is indeed helpful. So we will compare with a setting where all the model weights are simply randomly initialized. And also we want to understand like whether this fine-tuning as more data come in, we do actually update the embeddings based on the new observation as well. So we want to understand whether that's beneficial. So in comparison, we'll have a like competing model where the embedding of the GN learn parameter will be keep fixed through the iterative process. And lastly, we also want to compare with approach where we simply treat the embedding parameters as a free parameter. So this is kind of contrast with a graph neural network architecture. So in this results, we compare all those benchmarks that like ablation study I mentioned. As you can see here, we do see like a marginal like improvement from the model that initially like has a supervised training compared to the random initialization, but that difference becomes negligible. And then from the plot, I think one of the very important ingredient is that we do fine-tune the embedding as more data come in. And also the gap between the ensemble model versus the base model does show that like one of the key ingredient is that we have an improved predictive model that can improve the overall performance. And with that, I will mention a couple future directions that we're currently working on. So one is on the graph neural network side. So currently our model is actually nodes agnostic. We're not really using the nodes feature information, which could be useful because otherwise the model cannot distinguish like different nodes that has very similar local topology on the graph. And also due to like some computation constraint, we'll be interested in develop a neighbor sampling approach, which will enable us to use a larger subset of graph from the spoke feature. And also, as I pointed out earlier, that the improved predictive model does seems to be a very important part of the success of the model. So we're interested to investigate like more diverse set of model because current ensemble model is using all the same architecture, but they only differ by like random different initialization. And then lastly, there's also interesting to develop instead of a co-star where you randomly select number of genes, maybe there's like efforts can be put there using like existing LLM or like different literature, try to have a better starting points than just random initializations. With that, I would like to thank you for your time. Also appreciate the collaboration with UCF and the colleagues from the National Lab. And then this work won't be possible with the general support by the LDRD funding from the lab. Yeah, thank you.